Well, good afternoon, everyone. So firstly, I want to thank Promega for providing this opportunity for me to speak today. So my name is Dr. Pamela Ward, and I'm in the Department of Molecular Pathology at the University of Southern California. And this morning, I'm going to present a brief presentation, and the title is Exploring Microsatellite Instability Screening Technologies. And so we all know that we're in the midst of immunotherapy era, and this has created a paradigm shift in microsatellite instability testing. And unlike no other molecular test, microsatellite instability testing has been driven by guidelines since 1997. And so what I want to do today is to give a brief review of microsatellite instability and then focus on how we perform that testing in my laboratory. And so these are my disclosures for today. And so let's start by looking at the guidelines that are out there. So here in the UK, since 2017, universal screening has been recommended either by IHC or by MSI to screen Lynch syndrome in colorectal cancer cases. And then last October, this was extended to include endometrial cancer, but the recommendation was to do this only by IHC. So then jumping over the channel, let's moving into Europe. So here, the European Medicine Agency has made recommendation to approve Pembro as first-line option for metastatic colorectal cancer patients that have microsatellite high instability or show mismatch repair deficiencies. And we expect this final decision to come out anytime in this first quarter. And similarly, here in the UK, the National Institutes of Care and Health Excellence are also developing an indication for MSI and mismatch repair indications for checkpoint inhibitor therapy. So if we go back to Europe, then ESMO have actually gone one step further. So in order to assess eligibility for treatment of these immune checkpoint inhibitor therapies, they have recommended that we use both mismatch repair IHC and MSI PCR. So here in the States, we have a mosaic health system, but we do have a National Comprehensive Cancer Network. And just recently, they've updated their guidelines for Lynch syndrome. And now the recommendation is for universal screening for all colorectal and endometrial cancers for the identification of Lynch syndrome carriers, regardless of age of diagnosis. And this can either be achieved by MSI and or by IHC. And then in terms of FDA approvals that we have over here, just last year, Pembro was now approved for first-line treatment of advanced cancers that have microsatellite instability high or deficient mismatch repair. And at the same time, Pembro is now approved as a second-line treatment for unresectable metastatic tumors with a high TMB, TMB being tumor mutational burden, but we want to see at least 10 mutations per megabase. But the caveat here is that you use a companion diagnostic test that is put out by Foundation Medicine. So what actually is a microsatellite? So it's a DNA motif that can be repeated anywhere from 5 to 50 times. And within this motif, we see repeat units of 1 to 6 base pair units. And these microsatellites are found throughout the human genome. And specifically, for microsatellite instability testing, we exploit mononucleotide repeats. And here's an example of a mononucleotide repeat, BAT26. And so BAT26 is 26 anucleotides located in exon 5 of the MSH2 gene. And BAT, B-A-T, stands for big adenine tract. And so what is instability? So during DNA replication through a repetitive sequence, the polymerase complex can actually lose fidelity and can slip such that a strand loop is generated. Now, if that loop occurs on the newly synthesized strand, then we get an additional A incorporated into that new strand. Alternatively, if we get that loop generated on the template strand, then with the new strand, we have an A nucleotide omitted. So we have the result of a missing A. So these two anomalies lead to insertion and deletion errors. So because we have a high number of these replicated sequences throughout our genome, we need to have an elegant mismatch repair system that can actually identify and correct these DNA mismatches. And so this system is comprised of two heterodimers. And so we have MLH1 complexing with PMS2, and we have MSH2 complexing with MSH6. And if this system is compromised in any way, then that leads to the accumulation of replication errors, and obviously this increases the risk of cancer. Now, if we see that these errors occur in the germline situation, so it's a genetic hereditary condition, then this is obviously Lynch syndrome. Alternatively, if we have MLH1 methylation, then this is where we see sporadic deficiency of mismatch repair. Now, both of these anomalies together generate tumors that harbor a high number of mutations. And these high number of mutations lead to a high level expression of neoantigens. And this is the reason that these tumors are highly responsive to immunotherapy. And then from a testing perspective, Foundation Medicine coined the term tumor mutational burden. And a TMB is considered high, as I've said previously, when we have at least 10 mutations per megabase of DNA. So the gold standard for determination of MSI is actually to use PCR followed by fragment analysis. And a number of labs, in fact, a good number of labs use the Promega kit, which has been available since 2004. And this kit assay comprises of a multiplex assay where the reaction contains primers towards these 5 biomarkers. So we've got BAT25, BAT26, NR21, NR24, and Mono27. So BAT, again, is big adenine tract. NR simply new repeats. And Mono is a mononucleotide repeat. So that's where the nomenclature comes from. And so all of the work that I'm going to present in the slides that are hereafter, we've been using the Promega MSI kit version 1.2. But obviously now their OncoMate kit is available. It contains these same biomarkers, and this kit is actually already CE IVD labeled in Europe. So in order to do this test, we obviously have to isolate DNA from both normal and tumor. And these are the conditions under which we validated this test in my laboratory. So we want to have a tumor section that has at least 30% minimum tumor content. And then our anatomic pathologist will go ahead and circle areas for us to extract. So we will circle, he circles the tumor in red and then normal in blue. And then this goes into the lab and the techs will perform macro dissection. We actually use the Promega FFPE DNA isolation kit to recover the DNA. We then determine the concentration by nanodrop. And then going into PCR, we want to make sure that our DNA concentration is between 6 and 20 nanograms per microliter. If the DNA concentration falls within this range, we will always get a result. It's never failed. So then taking the DNA of this concentration, we put 2 microliters into the PCR. Following PCR, then we do capillary electrophoresis. So there's a standard condition for electrophoresis, but we actually perform 3 different injections. So we use the normal, which is what's indicated on the package insert or in the Promega protocol. We then do an extended injection, and then we do a reduced. So we want 3 different amounts of basically DNA being analyzed by the capillary electrophoresis instrument. And from there, we're going to mix and match in the analysis because we want to see balanced peak heights in normal and tumor. So what do I mean by that? So if you, so here we have two ferrograms taken from tumor. So the tumor is black and normal is the green. And if you overlay those ferrograms, you can see that we've got matching peak heights. Because if we have a subtle case of microsatellite instability, that it's important to interrogate the edges of the stutter profile. And so in order to do that accurately, you need to have these balanced peak heights. So the predominant peaks are pretty much matching. So typically here is what you'll see how microsatellite instability results are presented in papers and at presentations. And so this is from a colorectal cancer case. And the top panel we have DNA from normal, and in the lower panel we have DNA from a tumor. So in each, you can see these stutter profiles are generated in each of these 5 biomarkers. Now, as we get replication errors in vivo, then we get replication errors in PCR. And obviously these errors can't be repaired. And so each peak represents a PCR fragment that has either lost an A nucleotide or has gained an A nucleotide. And that's why we get this stutter profile from PCR through these mononucleotide repeats. If you don't see a stutter profile, then you know it's an artifact. And in fact, we routinely get an artifact coming up with NR21. You can see it have an asterisk over that single peak. So that is an artifact. That has nothing to do with microsatellite instability. And so now looking down at the lower panel, this is DNA from the tumor. And so tumor is obviously always going to be a mixture of tumor and normal DNA. So in the case of microsatellite instability high, we'll see 2 sets of stutter peaks. So if each of the biomarkers on the lower panel, if you look at the stutter profiles to the right, you'll see that they're matching up with normal, and then the novel tumor stutter profiles are shifted to the left. And invariably with a mononucleotide repeat, we always see a shift to the left. But that's not how we analyze the DNA in my laboratory. What we actually do is to overlay those ferrograms. And so in the bottom panel, you can see that we've now overlaid the top panel normal onto the middle panel tumor. And so now it's very easy to determine the normal component of the tumor and then to look for the novel nucleotides base pair shifts in the tumor DNA. So in colorectal cancer, we often get these profound shifts. And so it's not actually necessary, I guess, in this situation to actually overlay these, but this is the process that we use, and I'll explain why in a minute. So if we look here in this particular case, we've got a 4 base pair shift in the novel tumor in NR24 marker, and if we look all the way to the left, then in BAT25, we've actually got the novel allele now, 12 base pair shift from normal. And see that these are clearly easy to determine, and every biomarker has microsatellite instability, and so this is microsatellite high, instability high. However, this isn't always the case, particularly when we start to look at extracolonic tumors and specifically endometrial. And so we sort of look and categorize our stutter shifts. And so the panel to the left is where we have more than a 2 base pair shift, and this is routinely a pattern that you see in colorectal. And again, it's absolutely unequivocal. It's definitive MSI. We have novel peaks in the tumor represented to the left. But let's focus on the center panel where we've got a subtle MSI. We do actually call this MSI, but there's a subtle shift of the predominant peak either by 1 or 2 base pairs. So in this particular image that I've got here, you can see that the predominant peak is shifted by -1. So we've got a -1 base pair shift from the tumor, which is red, and black, which is a normal. And so in order to call MSI, we want to see a shift in the predominant peak, but we also want to see additional stutters in the direction of that shift. So if you just look, you can see additional peaks in the red profile, which is the tumor. So to us, this then indicates that this is a definitive shift. We've got a shift in the predominant peak. We've got additional stutters to the left. But the only way you can interpret this data is to have balanced peak heights. Otherwise, you're going to be missing peak data and having to sort of estimate what you think might be the image at the leading edge. So this is why we have those 3 different injections where we mix and match so that our final image, as you see here, the predominant peaks are matching in peak height. So it's important to actually look for those additional stutters to the left, because if we look at the image panel now on the right, this is a biomarker, NR24, that's been taken from a microsatellite stable tumor. And you can actually see, so again, the red is tumor DNA and black is normal. So you can clearly see that we have a 1 base pair shift in the predominant peak height in the tumor. The tumor is -1 compared to normal. But then if you look to the left, there are no additional stutter peaks in the tumor. And so this is MSS. Quite often we'll have a biomarker where the tumor DNA and the normal DNA will show heterogeneity. So in this case, we have one allele that's got 24 A repeats, and in the other allele we have 23 repeats. And during PCR, it's sort of a random event as to which allele gains PCR advantage and is amplified preferentially. And so we do see this flip-flop between peak heights. And we know this because from our validation experiments where we do replication precision, where we repeat the same sample multiple times, this is where we see that these peaks can vary by plus or minus 1 base pair, which generally indicates, as here, we've got a heterozygosity of the marker. So, So just to further clarify, in the center here, if we see a 1 or 2 base pair shift where we get these additional stutters, we always call this MSI. This is when I first started to work with microsatellite instability. We were doing validation work with the Mayo Clinic, and that was their standard, to use a 1 base pair shift as MSI. And so that's a standard that I've taken, and it's a standard that is also given through the College of American Pathology when we do our proficiency surveys. And so just to show you how subtle MSI can look in endometrial cancer, so this is actually a case. And so we have these 5 biomarkers. The red is the stutter profile from tumor, and the green are the stutter profiles from normal. And let's look. So we have NR21. So we do have a predominant base pair shift, but there's no additional stutters to the left. So this is MSS. And the next 3 markers, we have base pair shift, and we see additional stutters to the left. So each of these are called MSI. And then mono 27 on the right is actually equivocal, because even though there's a predominant base pair shift and there seems to be evidence of additional material in tumor moving to the left, we don't actually see a new novel stutter peak in tumor versus normal. And so we've called this one equivocal. But in order to call something MSI, we need at least 2 biomarkers to show instability. Here we've got 3, so clearly this tumor has a phenotype of MSI high. And so I give them the reference at the bottom. MSI high, the definition by fragment analysis, if we have less than 30 biomarkers that show instability, then that has a designation of MSI low, microsatellite instability low. If we have more than 30% of those markers showing instability, then that's microsatellite instability high. And determining MSI or determining mismatch repair errors by PCR, we have a sensitivity of 95%. So obviously we can look at the actual mismatch repair genes to look for mismatch repair deficiency by immunohistochemistry. And so here we're looking for a loss of MLH1 plus PMS2 or MSH2 and MSH6, which then will indicate that we have mismatch repair deficiency. And similar to MSI PCR, the sensitivity of IHC is approximately 95%. Now, there's discordance between IHC and MSI such that both technologies miss about 5% of cases. And so the actual recommendation is to do co-testing, so we don't miss a single case. So every patient that's eligible for these immunotherapies has the advantage of being put onto those protocols. So of course we can also determine instability, mismatch repair by NGS approaches. However, the approach has a lower sensitivity, it's 85%. Obviously, there's an enormous cost difference, and turnaround time is obviously impacted when you do an NGS. So even though you can achieve looking for mismatch repair errors by NGS, it's quicker, cheaper, and actually a little bit more sensitive to do it by IHC and PCR, in which case you can perhaps approach 100%, which is obviously favorable for the patient. So measuring response to immunotherapy, or in fact to any therapy, we're looking for a RECIST score, which is response evaluation criteria in standard tumors. And this is made by taking longitudinal measurements of tumors from radiographic images. So is there a way now that we're in the era of circulating cell-free DNA and liquid biopsies, so is there a way that we can track microsatellite instability status from a liquid biopsy sample of a patient on immunotherapy? And so we've actually done a small pilot study. These studies are continuing, but I just want to briefly show you the result of one patient, just to show you the potential of perhaps where this kind of work can lead us. And so we have a stage 4 colorectal cancer patient, and they're on immunotherapy. And this is just one biomarker taken from that tumor. So this is the primary tumor, biomarker NR24. We have the red profile from the tumor, and we have the black from the normal of the tumor. And then we've taken a liquid biopsy sample, and so this lower panel is the result of that biopsy, liquid biopsy. So from that biopsy, we recover, isolate circulating cell-free DNA. And then we also collect the buffer coat and extract DNA from the buffer coat, so we have genomic DNA. So the genomic DNA buffer coat is black, so that's normal. And then the red is the circulating cell-free DNA. And so likewise, as in tumor, circulating cell-free DNA from a stage 4 cancer patient is going to contain both normal and tumor DNA. And so very easily you can see in this lower panel where we have the novel stutter profiles of the tumor shifting to the left. We also see red stutter profiles to the right, overlaying perfectly with normal. And if you look at the top panel of the tumor and the lower panel with the circulating cell-free DNA, you can see that we're looking at the same tumor cells here. We have the same heterogeneity of the biomarker NR24 in the tumor, where we've got two different events that are represented here, because we've got two prominent peaks. And so now looking at results from this patient, so we've got the initial biopsy taken at baseline, and then we've got biopsies taken at 4 weeks and then at 6 months later. So looking at this first panel, this is where we've got circulating tumor DNA versus buffer coat DNA. So MSI PCR, it's a qualitative answer. It tells us yes or no. But to some degree, this analysis is semi-quantitative because we can look at the peak heights and that can be influenced by how much DNA we're loading into the electrophoresis. So it's not an accurate measure of quantitation, of course, by any means, but a semi-quantitation means I think it's quite good in it, and it very clearly shows here that if we look at these arrows in the first biopsy panel, that more or less equal peak heights between the normal DNA from the liquid biopsy versus the tumor. So here we consider there's a 1-to-1 ratio of normal to tumor DNA in the circulation. Now let's look at the center panel. And so this is a biopsy now taken 4 weeks later. The patient is on immunotherapy. We know from RECIST data that the patient is responding to therapy. And now look at the peak heights. So again, we're focusing on the red because the red is circulating cell-free DNA. The starter peaks to the left are tumor, and those on the right overlaying buffer coat are normal. And so you now can see a profound difference in the peak heights. So we have 4 times less tumor DNA than we do normal DNA. And so this is an indication that there's less circulating tumor DNA, which is indicating that the patient presumably is responding to immunotherapy. And then quite remarkably, by 6 months, we know the patient is still responding. We're looking at the panel on the right, and now there is no evidence of circulating tumor DNA. We only see circulating normal DNA, which is matching exactly with the buffer coat, the black profile. And so we're very encouraged by this data. We have data from 12 patients currently, and the study is continuing to see, just the potential of where this kind of work can lead us. And so, in conclusion, then, what I've done today is I've spoken about MSI PCR fragment analysis being the gold standard. I've described how we've achieved that validation and what standards we use in my laboratory. We also overlay the ferrograms so we can detect subtle shifts. And this is exceptionally important if we want to start and use these panels to look at extracolonic and specifically endometrial cancers. We know that fragment PCR, MSI PCR fragment analysis detection has a sensitivity of 95%, likewise immunohistochemistry, and so the recommendation, so that we don't miss a single patient who can qualify for these immunotherapies, we recommend co-testing with MSI and IHC, and we're beginning to look at how we can exploit liquid biopsies, circulating cell-free DNA, to look for MSI status to track response to IO, and these are ongoing studies. And so thank you for your attention, and I'll be happy to take any questions.